Okay, so this is going to be Kidney Podcast 2.5 to be done with part two as your homework over the weekend. So, taking a look again, so kind of quick review, filtration's occurring between the glomerulus and the Bowman's capsule. So pretty much everything but the plasma proteins are going to filter out at this point through those very porous walls of the glomerulus, and that filters into the Bowman's capsule region. So then it's going to travel into this proximal convoluted tubule. So, a normal capillary would filter about, well, not very much through it. They're pretty tiny and not much filtration goes on. These capillaries are going to filter about 20% of your actual total blood volume. So they're definitely more filtration going on there. So the average amount of glomerular filtrate produced per day is about 180 liters. And so, remember we've talked about your total blood volume is about 5 liters. So you're making way more glomerular filtrate than you even have liquid in your blood. But the average amount of urine produced each day is only about 1.5 liters. So the big question is, how is this possible? So we filtered it all out, so forth. How are we going to get a lot of this good stuff back? Because it's not very particular about the way it filters in the first process. So luckily there's 2 other processes in our kidneys that function to bring back a good substantial portion and the particles of all of the things that came out. So one key component of our kidneys is the fact that they are functioning in osmoregulation. So that's controlling the water balance of the blood, the tissues, and the cytoplasm of a living organism. So our kidneys really are responsible for aiding in our osmoregulation or our water balance. And then there's some key hormones like antidiuretic hormone that are also going to do or help with that, and that comes from the brain. So the next piece, the big piece that's occurring in our proximal convoluted tubule, and the reason for those microvilli is reabsorption. So almost all of the filtrate that we've taken out must be reabsorbed back into our bloodstream, or we would dehydrate extremely quickly, lose a lot of our nutrients and so forth all through our urine. So again, this occurs mostly in the proximal convoluted tubule. So proximal, the closest to the point of attachment. Convoluted, kind of, wavy, in a tubule. So some of the things that are reabsorbed. So we start off with our sodium ions. They move out by active transport. Chloride ions are always going to follow. So here's our positive ions, our sodium. Our chlorides are negative, so they're going to follow to balance out that the ionic balance, or that difference. So chloride ions follow the sodium ions. Glucose molecules are co-transported out of the filtrate into the fluid outside the tubule, and eventually those are going to re-enter the bloodstream. And this occurs by co-transporter proteins in the outer membrane of the tubule cells. So it's just a different kind of protein. And then other molecules that are reabsorbed are amino acids, mineral ions, small proteins, and water. So again, the majority of this is happening in that proximal convoluted tubule. This video that's showing on the screen right here, I actually placed on the website. So as you're going through these different podcasts, if as you finish up, the videos really kind of help to solidify some of those different concepts. So we've talked about, our filtration happening here, our reabsorption happening here, and then a really key thing occurs in our loop of Henle, and this is in balancing our water amounts. So the job of the loop of Henle is to make the tissue fluid in the medulla hypertonic. Remember diffusion concepts? So hypertonic, it has more dissolved substances in the fluid than what you're comparing it to. So more dissolved substances, less water compared to the filtrate in the nephron. And this is going to help to reabsorb water. So the way it does this is the first part of our loop of Henle is thin. The walls are thin, but they're impermeable to sodium ions. So there's no sodium channels in our descending loop. And then our ascending loop is impermeable to water, but it has sodium channels, so it actively pumps out excuse me, sodium chloride. And so normally water would follow by osmosis, but it can't do that. So, so it kind of sets up this really unique kind of a system of concentrating our sodium ions as it goes farther down the loop of Henle. And we'll talk a little more about basically how this works a little later. What happened there? So I think we'll stop there. So have a good evening.